Hi, welcome back. Today we'll be talking about targeted HiFi approaches with PacBio. In our previous video, we discussed the power of PacBio HiFi sequencing for whole genome analysis. But what if you're interested in specific genes or regions? Targeted sequencing can be a highly effective strategy. So, why choose a targeted approach? Often, whole genome sequencing provides more data than you might need, and it can be less cost-effective, especially when you want to sequence large numbers of samples. Targeted HiFi sequencing allows you to focus your sequencing power directly on the regions of interest. This makes projects more scalable and affordable, whether you're using a benchtop Vega or higher-throughput Revio system. Targeted HiFi brings significant advantages. Just like with whole genome sequencing, it allows for robust coverage of difficult-to-sequence regions. Think of tandem repeats, genes within segmental duplications, or those with highly similar pseudogenes or paralogs. These are areas often challenging for short reads. The long, accurate HiFi reads enable unambiguous haplotype phasing directly from the data, without needing parental samples, trios, or statistical imputation. Plus, you get high-accuracy variant calling, including complex structural variants, and copy number variations within your target regions. Many labs want to consolidate and modernize their workflows using NGS of exomes or genomes. However, not all regions of the genome are accessible to NGS, so labs often find themselves stuck running older, laborious molecular workflows. These can include Southern blotting, MLPA, qPCR, or reflex Sanger sequencing for ambiguous results. For these cases, long-read targeted panels can be used to consolidate multiple legacy workflows. This is incredibly valuable in clinical research, high-throughput settings, or production environments where consistency and efficiency are absolutely key. PacBio offers 3 main approaches for targeted HiFi sequencing, each suited for different applications. First up is PureTarget. This is a PCR-free method that uses CRISPR-Cas9 technology for enrichment. Because it avoids PCR, it preserves native DNA methylation and performs well even in hard-to-amplify regions, like those with high GC content. It's particularly well-suited for studying repeat expansions or when the methylation status is critical. PacBio even offers an off-the-shelf PureTarget repeat expansion panel targeting 20 neurological disease-related genes. Second, we have HiFi target enrichment, which uses long-read optimized hybridization capture panels. This method is excellent for creating small or large gene panels when you need comprehensive coverage across your targets. This includes calling structural variants and phasing haplotypes. We partner with companies like Twist Biosciences to offer off-the-shelf panels, such as dark genes or pharmacogenomics, and also support custom panel designs through our partners. The third approach is sequencing PCR amplicons. If you can amplify your target region using long-range PCR, PacBio HiFi can sequence the full-length amplicon with high accuracy. This is a very cost-effective and straightforward method, great for smaller gene panels, validating specific variants, or replacing older Sanger sequencing assays. Customers typically design and optimize their own PCR primers, and PacBio provides solutions for indexing those samples. Choosing the right method really depends on your specific research question and the nature of your targets. You'll also want to consider whether you need methylation data, the size of your panel, and your budget. PacBio provides protocols and support for all 3 workflows. By leveraging targeted HiFi sequencing, you can gain deep insights into specific genomic regions efficiently and cost-effectively. You'll even be able to tackle challenging targets that were previously difficult or even impossible to analyze accurately.